Dámy a pánové, ladies and gentlemen, good morning. Přeji vám krásné ráno. Sešli jsme se zde v rámci druhého dne z Mendelových dnů, které jsou letos věnovány tématu Mendel a svět. Připomínáme si 200. výročí od Mendelova narození a v rámci projektu Interreg se scházíme. Jsem velice ráda, že se zde setkáváme taky s našimi partnery z Rakouska, z Konrad Lorenz Institute, z Vídeňské univerzity a podařilo se nám také navázat nová partnerství s Naturhistorisches Museum. Jsem velice ráda, že zde můžu přivítat také pana Petra Šímra, Linčiu z Konrad Lorenz Institute, Petra Šímra from Naturhistorisches Museum, Barbara Fischer z Vídeňské univerzity a z Konrad Lorenz Institute, naši partneři. Samozřejmě zde vítám taky všechny další účastníky z opatství a ze firmy Společně a všechny vás z Masarykovy univerzity a další hosty. Dnešní téma je tedy workshop Teaching Mendel and Genetics. Ráda bych zde vlastně sdílela s vámi naše pracoviště Mendelova muzea, poznatky, jakým způsobem vzděláváme jak studenty, tak návštěvníky muzea. Bude velice pěkné, že jsme se sešli z různých institucí, jak z univerzity, tak z muzeí a jakým způsobem v dnešní době přistupovat k popularizaci vědy a Mendela. Takže budu ráda, když budeme takhle tyto naše zkušenosti vzájemně sdílet. A jako první zazní příspěvek Linčiu, která pohovoří na téma Philosophy of Science in Informal Science Spaces, Engaging with the Science of Inheritance, Deck 50, Vienna. Because Ines Mehu-Blantar from Naturhistorisches Museum wasn't able to come today, Lynn will be speaking also with Peter Schimmer about and explaining her presentation as well, presenting also her presentation. So Lynn, microphone is yours. Thank you. Okay.
Hello, everyone. Thank you, Blinka, for the wonderful introduction. So, as was mentioned, we represent the University of Vienna side of this overall Interreg project, Gregor Mendel's Legacy for Science, Culture, and Humanity. And I'm really quite thrilled today because for our part of the presentation, we have a three-part little workshop here, which is between the head of the Deck 50 Immersive Science Center at the Natural History Museum, Vienna, and us as the scientists at University of Vienna bringing a project into this science space. And we have Peter Zimmer, he is the anchor of the Natural History Museum, a long-term science educator and science communicator at the museum who is managing a lot of and running a lot of our projects in person at the Deck 50. So with this three-part presentation, our hope in this workshop format is to have an exchange between the three of us about how different perspectives come in. The person who is organizing the space, the person who is an outsider bringing in a project to the space, and the people who are actually running the project on the ground. Unfortunately, Ines couldn't be with us today, so Peter will present her slides. So we will have first me giving this little general overview of the three of us, and then Peter will talk about Ines's part, which is what the Deck 50 is, and how it will, what its facilities are and how it will be used. And I will talk about how we bring the project in using the capacities of the space and the formats that Ines has developed for the space. And then Peter will finally enlighten us on how it actually went. And in addition to that, to talk about all the other types of projects that he has been working on at the museum in science education, some related to genetics, some not. So I also appreciate this opportunity, organizing this workshop so that we can exchange with our colleagues at Masaryk University and to see how things are run in the science space here at the Mendel Museum. And I'm also grateful for some of our speakers from yesterday to be here, experts on Mendel. We are not historians of Mendel, we are not experts here. So it will also be wonderful to hear from you how our parts work on the informal science space. So my background is in philosophy of science, philosophy of biology, and also in evolutionary biology. I'm in the evolutionary biology department right now. Our team, this is part of our team. Barbara Fisher and I are co-running the University of Vienna side of the project, and we have recruited a lot of different kinds of science communicators to work on this project together. In Mendel garb, on the bottom left, is Severin and Gernot. They are master students who impersonated Gregor Mendel in our interactive theater program that we will talk about soon. And Katrin is a designer who worked with us on our graphics and part of our art science game that we will also talk about. And Maria is a videographer and YouTuber, and she's the one who provided us with our film footage. So this is, I will soon give the floor to Peter, but I will just leave you with this slide first, is that Deck 50 is a brand new space that opened during the pandemic. So we were the first externally funded program to come in with the money and the resource and the scientists to develop programs in this space. They already have other programs that are running, and Peter will tell you more about that. This is the team that we are working with and the lovely trainers on the ground that are carrying out our program. So this is really a multi-level collaboration here. All right, Peter, would you like to help explain what the Deck 50 is and Enos's part?
So, hello, good morning, everybody. Thank you very much to Masaryk University for this invitation for this very interesting event. And I'm going to tell you now something about Deck 50. Since my colleague Ines Mehu-Blanchard is ill since yesterday and she unfortunately couldn't come, and so she asked me to do the talk for her. So Deck 50 is located at the second floor of the Natural History Museum. The Natural History Museum, one of the 5 largest museums of its kind in the world. Some 80 scientists are fully employed at the museum, and there are collaborations with many institutions like the University of Vienna, for example. Museums bridge the gap between visitors and scientists. That's why you can see this image of Golden Gate Bridge here. And Deck 50 is a very large modern hall that was only opened by the end of 2021, thanks to the pandemic 2 years later than originally intended. And it's really a huge hall with some 260 square meters, with a lab area, with an encounter zone, with an area upstairs, the red area, where children can also take part in the workshops. And there are open events, there are lectures, congresses, school formates. So lots of things to do and to encounter. It's very interesting and important that it's a hall designed for many different generations, for scientists as well as for school children. So it's a interlocking dialogue between different groups of persons and also different objects, real objects like taxidermy animals that we have normally at the museum, and also digital formates. Yeah, here you can see the wolf that represents the station about the wolf in Austria. And it's also important that there have been done interviews with collaborators of the museum, with visitors, how they see the present problem with the wolf in Austria, because since 2016 we have a very small reproducing population of wolves in Austria. And unfortunately, not to everybody these wolves are welcome. So it's interesting and important to talk about the consequences and the whole project. And some of these data that are collected by the visitors are also interpreted scientifically via social sciences afterwards. As I mentioned, we have two rooms or two compartments for school projects, the so-called white area, where we use microscopes, and the open lab, for example. Microscopes are always highly welcomed by school children, whether they have much experience or not, but microscopy is always very, very interesting. And upstairs, the red area, which is more confined for handicrafts, for the more relaxing part of school projects, and you will see later on what came out of this. And then there are the public lectures, place for stakeholder engagement, public lectures for visitors, but also the format that's called living with biodiversity. Regularly, scientists visit DEC 50 to speak about their scientific results, to give lectures either to colleagues from the museum or to the visitors. It's normally rather short lectures, about 20 minutes, and there is about 20 minutes time for discussions and for questions. This is part of our Mendel project when DNA was made visible for the children. And we have also this format that's called Meet a Scientist. This is colleague Severin from the university, whom Lynn has mentioned already, who was acting as Gregor Mendel, and is part of the Interreg project. Vielfalt kennenlernen, how to know biodiversity. That's a very important project that we are running since the beginning, since the opening of DEC 50, with various groups of animals, butterflies, spiders, dragonflies, hymenopterans, so that's bees and wasps and ants. And we have a little video here. Children have to try to identify these various diurnal butterflies with real objects and as well with digital tools. And the opening section with my colleague Harald Krenn from the university, and also in collaboration with scientists from the Natural History Museum, from the Butterfly Collection, and the very skilled ones could do origami specimens. And there was also the microtheater. About the microtheater, I'm going to talk a little bit later. The open labs are open for the visitor that's coming just out of personal interest. We know that about 60 to 70% of the visitors are coming with their families or with friends. Only about 5% of the visitors are coming alone, and the rest are coming with school projects. And so it's very important to have a format that's interesting for families, especially in the weekends. That's called the open lab activities. And there is always the possibility to discuss things, to ask questions to the scientists, and also to do some hands-on programs. So we had a genetic biodiversity workshop in the form of the open lab, not just for school programs, the Drosophila workshop, where the participants could actually detect various mutants of Drosophila, of the little fruit flies, in a dish, and also do DNA extractions. And it's very interesting to actually see the DNA then, whether you do it with tomatoes, for example, or other fruits. It's not very, very complicated. And this microscope, this research microscope, is the one that we use for the microtheater. That's a presentation of small animals, plants, objects, natural objects, onto the huge 12 square meters LED screen. And then the visitors can experience these small creatures or plants. So the programs of the Open Lab and the microtheater, together with a dinosaur presentation, is given mainly in the weekend and during the holidays, because it's open for the visitors. Especially the dinosaurs are really a hit for the children, as expected. But I'm very happy that we have so many different formats in this very modern hall. And yeah, good luck, Gregor Mendel, and good luck with the future projects. Thank you very much.
Thank you for introducing the Deck 50. So I will try to use this mic. Is it working? Okay. So now I will share my part, which is as an outside group from the Department of Evolutionary Biology at University of Vienna. How did we work with the format of Deck 50, and how did we bring in our project under the context of our Interreg program? So these are our goals, because we are an interdisciplinary team of evolutionary biologists and philosophers and science communicators. We have both scientific and philosophical goals that we like to achieve in our program. So the scientific and / philosophy of biology goal is to tackle the following issue, which is we are in a program that is commemorating Gregor Mendel's 200th birthday. But it is also 200 years from his birth date. So how can we introduce the updated science of inheritance in this context to 14 to 17-year-olds with Mendelian genetics as one type of inheritance? So the philosophical issue here is that inheritance is a much broader concept than genetic inheritance, which is much broader than Mendelian genetics. So how can we introduce the science of inheritance with Mendelian genetics as a part of it? The philosophy of science go is another way that we can address the issue of commemorating Gregor Mendel in modern times. So we can emphasize the scientific process of how he reasoned through his experiments. We can talk about the process of becoming a scientist. These are all inspirational topics for students of this age. And then we can talk about, why do science at all? What does it mean to do science? Why would anybody want to be a scientist? Through his story. And then we can talk about the usefulness and limitations of idealized models with Mendel as an example. So these are our goals. So in terms of scientific goals, we are influenced by papers that have been talking about why is it that we teach Mendelian genetics as a mandatory part of our middle school, high school education, but then we just stop there. So Gregory Radic advocates for teaching students the biology of their time, arguing how Mendel's legacy could hold back the teaching of science if we only teach Mendel and stop at Mendel. Working with Annie Jamieson, who is now also working in the museum world, they talk about how this genetic curriculum that we have now can contribute to entrenchment of genetic determinism, thinking that genes are the essence of life, for example. Kostas Kamparakis, he even argued for a Mendel-free biology curriculum, which is a very radical, provocative approach. So, but in light of these issues, we wonder if for a Mendel program, we can talk about inheritance more broadly and introduce Mendel in the context of a broader conception of inheritance. So we are influenced by work that have been addressing epigenetic inheritance, for example, behavioral inheritance, ecological inheritance, different modes of inheritance, developmental genetics, and Mendelian genetics as part of genetic inheritance. So how can we introduce all of these different conceptions in the same program that is going to be fun and creative and engaging at the same time? We also address the concept, but not explicitly at the end, of the idea that the genome is reactive to its environment. It's the environment that makes genes express themselves. So how do we situate the genome in environmental contexts? So on the philosophical go, as mentioned before, we want to showcase the process of Mendel becoming a scientist, his journey, and the struggles and challenges of that journey, and then what he learned through that journey. We want to normalize the presence of failure in science. It is very important to acknowledge this, especially for inspiring new scientists. And the process of scientific reasoning, as well as the emotions that are involved in that: curiosity, discouragement, frustration, enthusiasm, the puzzling through of things, the aha moment. So this is what we want to build into the interactive theater program. And then the importance of his work with a supporting scientific community. As we know, Mendel is not a lone, isolated genius. He was in a very supportive community here. And then finally, how idealization simplifications in science in his work help promoted new scientific developments, but in turn will limit it if we continue to stick with the same assumptions. So this is the poster of our program. It is completely conducted in German because we are attracting Austrian schools, and we had enough funding to fund 20 school workshops and with the prioritization of marginalized schools that don't usually have access to the Natural History Museum, for example. So we developed 4 programs: a Meet a Scientist, Meet Mendel interactive theater, a microscope lab of staining chromosomes, a science quiz about broader conception of inheritance, and an art science game called Mendel's Fantastic Piece. So we'll go through this very quickly to talk very briefly about how we approach the design as partners bring program to the Deck 50, and then Peter can enlighten us about how it actually went at the end. So Meet Mendel. This is actually a twist on the format Deck 50 had for their Meet a Scientist program. So Meet a Scientist is a program whereby they invite real scientists to come and talk about their stories, about their scientific life, and as an inspiration for school students. And the audience gets to interact with these scientists with this QR code board. So what happens here is that throughout the presentation, there is a 10-block format of a script, so to say. And then the scientists can fill in the script with their stories, with their experiments. And then throughout the script, they will have these Q&A moments to engage with the audience. The audience would have these boards that they can raise up, A, B, C, D, and then the poll will be interactive and in real time showing up on the screen. So this, in a sense, forces the audience to interact with you. You don't just lecture to a very quiet crowd and then just be with it. So our twist to it is that, okay, how about we let Mendel be that scientist in the Meet the Scientist format? So let's meet Mendel. So we have our master students here impersonating Mendel, going through the 10-block script that we designed, and interacting with the audience as they raise up these QR codes. So we designed it in 3 acts. There's Act 1, Mendel joins the Abbey and becomes a scientist. Act 2, Mendel's experimental process, focused on his key paper. Act 3, Mendel reacts to current science, whereby we time travel him to the current days and then just show him the development of genetics since his passing and see how he reacts to that. And then we end with a take-home message. And our interactive questions include, do you think that a monk can be a scientist? With the implication of, a citizen science component here. Do you have to be a professor to become a scientist? What kind of people can become a scientist? What kind of training is required? So if Mendel wants to be a scientist, what does a monk need to carry out scientific experiments? They need a community, do they need funding, do they need equipment, so on. How would you have first tried to solve the scientific puzzle? This is about Mendel trying to figure something out and then asking the audience to generate some hypotheses for his work. and then analyzing these hypotheses, which one is the most likely, what laws govern the flower colors, and then at the end, who can be a scientist, really? So these are the interactive questions. So my part was to develop the 3 acts and then try to incorporate as many principles as we could, following what we talked about earlier about talking about the process of science, the process of becoming a scientist, the role of emotions and failure, and so on. And then I gave this to our 2 collaborators, who then developed the very detailed German script for their play. And so this is some pictures from one of the theater performances with Severin here. And what was really cool here was that after Mendel reacted to current science, Severin effectively sent Mendel back, took off his robe, and then presented himself as a budding scientist, as a master student who is working in the life sciences. And then the students also went crazy over this. It's like, oh, wow, so what is it like to want to become a scientist now? And so there was an hour-long engagement here, in addition to the engagement before, and we were quite surprised with the results. But so that is the standalone theater performance that is provided under the Meet a Scientist context. The other program is the school workshops, and this is when we invite the 20 schools to come in a 3-hour-long workshop whereby we go through these 3 types of activities: a lab activity in the creative space that's mentioned before, and and another activity under this quiz environment. So these are the 3 environments. With the quiz environments, this is also a format that Ines developed for scientists who want to work with the Deck 50. It is another 10-block format of going through different topics with different questions that are thrown out. So that's why it's called a science quiz, but it's a fun trivia kind of science quiz game. And so the topics we wanted to talk about are different examples of inheritance. There are multiple channels of inheritance, and we mentioned Mendelian traits. We mentioned traits that are environment sensitive. We mentioned inherited through like behavioral across generations, with the parents bringing their offspring to similar locations and they have similar diets or temperature exposures and thereby continue to acquire the same traits. We introduced them to non-Mendelian traits, to microbial inheritance, and some new work on small RNA inheritance. So with these different examples, for each example, we have a quiz. Like, oh, what do you think is happening in this case? We ask an interesting question about it. They respond to that, and then the instructors respond to their response. So this is then followed by a museum tour, whereby the trainers who are very well experienced with the different kinds of exhibitions that are already in the museum, they point out similar or the same cases in the museum to reinforce what they were exposed to in the science quiz. So this is the whole class, starting with the science quiz. Of course, we developed the different protocols and manuals for each component. This was done in a way so that any of the instructors, I think there are 7 that we worked with, would be able to use these manuals and get the scientific inspirations and ideas behind it, as well as what they're supposed to do at a particular block, and then carry it out. So after the science quiz is done, the class is then divided into 2. So one half goes to the lab to do chromosome staining. The other half goes upstairs to the creative space to play with the art science game. And so here, this is conducted in 2 parts. First, we teach them how to use the microscope by observing samples that are already stained. So these would be human cells as well as other cells. And then we look at the chromosomes therein that are already fixed. And then they get to cut a part of the root of an onion and then stain it themselves and observe it again. So that is the lab component. What about the art component? So the art component is Mendel's Fantastic Beasts. It's a place where they can be creative, they can use some of the ideas that they were exposed to before, and then they play a game. So the game here is a mission, it's a story, and I think it's important to develop a narrative or a story when you're playing a game to immerse into the atmosphere. So here we have also the purpose of some of Mendel's work here at the Abbey, which is the climate-related work, the agriculture-related work, are work that actually benefit the city and the region as well. So here, the little Mendels, their goal is to help villagers protect Mendel's peas against climate change challenges. So they're going to design a beast. They're going to write down the recipe. This part is the inheritance component because they then pass it down to the villagers so that they can replicate the beast later. So this beast has traits that are activated by environments and diets, but also Mendelian and non-Mendelian traits. And then as they play through the game, they're supposed to record how they put together the beast, and then they pass it down. So these are the pieces of the beast. We prototyped and designed these pieces so that they are informative. They are hints about where they're supposed to go, what kind of parts they are, but also abstract at the same time. We'll talk about this a little bit later. But it took us quite a bit of time to design these pieces. And then we developed rules for them. So some of these are, some of these traits follow a Mendelian rule. So you would draw cards, for example. If you have 2 small k cards, you get this part. If you have a big K, small k, or big K, then you get to choose one of these parts instead. Others follow developmental genetic rules. For example, here we have a sort of a Hox gene scenario whereby when you throw a die, it creates a point, it triggers a different developmental genetic circuit. So it could develop a circuit that creates a fin or legs or hooves, for example, or this type of wing or a bat kind of wing or a butterfly kind of wing. And then we have ecological inheritance whereby they get to choose what season the piece grow up in, and then that will determine their color. So they can put sand on the piece, or they can determine what kind of diet it has, what kind of microbes it's exposed to, and that would determine the paint or the color of the piece. And so they go through these different stations, play the different games, and then they pick up the pieces and put it together. And so these are some of the results of these pieces that follow roughly the intended plan of having the head in the front, the tail in the back, and so on. But here's something interesting that I want to note. So there were explicit rules about how to play this game, and then these are the rules that determine which pieces they pick. The implicit rule is that they will put the head in the head slot, they will put the tail in the tail slot, they will put the wings in the wing slot. But this rule was not made explicit. So it's a rule that can be exploited, and then they can change the rules. So these crazy beasts could come out of changing rules by putting the head into the tail space, for example, or putting the wings into the leg space. And so this is a type of innovation that actually reflects actual biology in that you can explore phenotype space within constraints. So this was a meta level of what the game achieved. But interestingly, because of this game was really popular amongst teenage boys. And this was something we did not expect at all. We thought that, so even the teachers, when they came here, they said, oh, we saw on the program that there was going to be an art craft game. And we were like, yeah, the boys are going to be very not impressed with that. And we will have to force them to do it. But instead, they went crazy. So in this one example, where I personally went to observe, they were howling at how funny it was to put the beast in unconventional ways. So this kind of, they think they were like, maybe they were found a little loophole in the game, and then they were breaking the rules, but they weren't actually. They were following the rules in another way. And so that was an interesting outcome for me. And of course, there are situations of a developmental failure when a phenotype just doesn't work at all. And the students just have to calmly accept that they didn't know how to put this together. And so to conclude here, what we did was we have an informal science space with its own strengths. For example, what kind of things it can offer, what kind of formats are already there for us. And then we use this to develop our program within its constraints. These constraints include how many instructors can actually be there, what kind of time do we have for these programs to run, how many materials can we actually afford. And then when we develop the science engagement activity, we try to incorporate lessons from history of science, from philosophy of science, and of course also the latest science in the context of commemorating a historical and very important scientific figure. So together, this is a wonderful opportunity to explore different ideas in science communication, science education, and philosophy, and be able to work with a very broad interdisciplinary team. So a future question, possible question to have is this. We have a team of scientists coming in and then prototyping with the structures of the space, but we also have the trainers on the ground that are constantly improvising and interpreting the materials. They have to do so because Peter will talk all about this, why they have to do so. There are a lot of constraints and dynamics on the ground. And then we have the students and teachers and how are they contributing to the project as well. So there are 4 different types of groups. Here we also have like the head and also the staff of the Deck 50. Within this process, a lot of things could change. So it wouldn't all go according to plan of the conceptualization of the scientists, for example. So what changed, why, and is it because there are pre-existing biological frameworks already there that are prohibiting some aspects to be explored or enhancing others? And how can we feed back to the top so that we can use these constraints to also lead to innovation in the informal science space? So many thanks to the Deck 50, to Iris Alt, the head of science communications, to Ines, who is the head of Deck 50, to Agnes, who is the head of pedagogy, who helped us organize all the school workshops, as well as the Deck 50 trainers and instructors. Now I will give the mic back to Peter, who will talk about how it actually was on the ground and the other projects that he's involved in.
So, I know that I know little, but is this enough? I know that I know little is actually, or I know that I know, I don't know how this works. Now I know. Okay. Sometimes learning very, very fast. I know that I know nothing is a saying by Socrates, the Greek philosopher, and when he said that this somehow was a proof of ultimate wisdom to see that we know, well, not nothing, but only very little. And that's exactly the daily situation we as interpreters at the Natural History Museum are in. So I've been asked to talk about my professional experience, and so I'm going to tell you, I'm not starting with my birth date, but I studied zoology and botany at the University of Vienna from 1980 to 1988, and from 1989 onwards, I'm working at the Natural History Museum, and I have, let's say, a second leg to stand on. That's the Volkshochschule Poly College, the evening school Poly College in Vienna, where I'm also working since 30 years already. But I'm a freelancer, and that's very good. I'm not fully employed, neither at the museum nor at the evening school, but it enables me to work with many other institutions like several national parks, National Park Donau, National Park Thaya Valley Podi, the High Alpine National Park, the Kalkalpen National Park. I contributed to programs in Poland, in Malawi, in Madeira, and now I'm even a lecturer at the University of Applied Arts in Vienna, and I'm working, if there is not a pandemic, as a guide to nature areas for interested clients, young and old ones. So that's quite interesting. And like this, I never really get bored, rather overworked. And there is a huge variety of different work, and that makes the things quite thrilling. So about my work at the Natural History Museum. As I've mentioned already, it's a very large museum. We hold currently some 30 million objects at the collections. About 10% of those are open to the public in the display halls. We have 50 display halls. And besides the University of Vienna, this is the second largest natural history research unit in Austria. Oops. I'm learning fast, but not fast enough. So my main tasks at the museum are giving guided tours. These guided tours last more or less 1 hour. School workshops like the one on the Mendel beasts, the micro-theater where we keep small animals, mainly insects, for a short time in small dishes like this young bug here and present them via the microscope onto the screen. We do that as an open lab activity, as an activity for visitors who are just dropping in, and as well as for schools. We are giving courses in microscopy, guided tours to the rooftop which have a more historical aspect. And I was also collaborating in inventing new pedagogic programs, and I also collaborated with other departments of the museum, like the botanical department, the bird collection, the anthropological department, among others. So the role genetic plays at the Natural History Museum, and especially at my or our work, is little. I'm sorry to disappoint you, but it's quite small. In 2009 and 10, we had the special exhibition on Darwin. That's where genetic played the first time a major role among the exhibitions. And then last year we had a very funny exhibition, the Dinosaurs. So that means which role do dinosaurs play in various movies, starting with the first movie of Gertie the Dinosaur from 1914 until to the most recent productions of Jurassic World. And of course there arose the question, is it possible to clone a dinosaur, and how is it done? I must disappoint you once more, it's not possible to clone a dinosaur. And probably if you look at this guy, we can be happy that it's impossible to do it. And we have a special department for genetics at the Natural History Museum, the Central Research Laboratories, with a DNA lab where projects are done, for example, on the brown bear in Austria, on the European wildcat, on crustaceans and dragonflies in Vienna, on mollusks in Vienna and in the Alps, about the otter, various birds of prey, lizards, and the Garacyprinids from Oman. So you see international cooperation also on the genetic level. Then there is the program called ABOL, the Austrian Barcode of Life, with the aim to barcode genetically all the creatures, all the species in Austria, and there are already more than 30,000 tissue and blood samples in the collection of the research laboratories. So why does genetic play such a little role then in exhibitions? It's difficult to exhibit genetics. There is not much to see. You can present beautiful butterflies and stuffed bears and dinosaur bones. That's all like, oh, wow, beautiful, thrilling, interesting. But with genetics, it's a bit difficult. So since a few weeks, we have new display cases on the history of the Natural History Museum, and there the DNA lab has got at least a small section to present genetics about the genome-wide alignment, for example, or the thermocycler that's used in the DNA laboratory, or here on the left side you can see the evolutionary tree of chordates, so of vertebrates and their closest relatives, which was done on the base of genetics. Then there is a special display case on this Gara fish from Oman. It's quite interesting. It's the same species. Once they are eyeless white cave animals, and the other ones are fully equipped with eyes and live in little brooks on the surface. And there are also lectures on molecular biology and genetics regularly, which can be visited either by colleagues from the museum, from other museums, from the universities, but also by interested private persons or students. Genetics at the Museum, Part 3, the evolution of mammals on a molecular and genetic basis. You know, these two old groups of marsupials and monotremates, but the four more modern groups of evolution, the Afrotheria, for example, which prove that elephants and elephant shrews are quite closely related, although the ones are very big and the other ones are rather small. And genetic biodiversity you can actually discuss in many places at the museum, but it's rarely done. For example, we have this wonderful display case with more than 20 different African antelope species. All taxidermy specimens. Some of them are very, very old, like this bongo, for example, and some are quite recent, like this addax antelope. And of course, you could use genetics in such a case, but it's normally more the fascination for the animals themselves than talking about genetics. The problems of small populations, like the very small populations of European wildcats and the grey wolf that we have in Austria currently, or small populations on tropical islands. I discuss these problems sometimes in special guided tours in the weekends. Or the rather modern chapter of can we, shall we, will we resurrect extinct species like the thylacine from Tasmania, or the North American passenger pigeon, or the North American Carolina parakeet. These three are specimens actually that are on permanent display in the halls of the museum. Yeah, and our activities at Deck 50, like one school class actually doing the microscopy work on permanent slides on stained nucleus. So what's about my colleagues? My colleague Jessica Österreicher was so kind to provide me with some photos. Thanks to her. Our team of almost 20 pedagogists consists mainly of biologists. There are also some prehistorians, geologists, paleontologists. So we studied at universities and later on started to work here at the museum. And we cover very diverse topics, from minerals, meteorites, archaeology, to all sorts of animals, the architecture and the history museum, and even genetics. So many people think, oh, this is a student job. Until a few years ago, I've been asked, oh, you are still a student. Well, now I've got white hair, so they don't ask me anymore if I'm a student or not. Or an early-aged student, maybe. But it is definitely not just a job for students. You need, in the best case, lots of experience, you need a good knowledge, you need lots of flexibility, because things can change from one day to the other. Since the museum is very, very large, it happens sometimes that some objects that were there yesterday are not there today anymore. Nobody informs us, because simply the museum is so big that you can't be informed about every minor change. So you have to be quite flexible. Sometimes it's the case that one hall is so crowded with people that you have to avoid this hall, which is quite different if you have to avoid the dinosaur hall, but you have a dinosaur guided tour, for example. And so you have to be flexible. We have always to talk to each other before the guided tour starts in the morning. Where are you going to start? Where are you going to start? And then I must say it is a real profession. It's not just some professional bridge that bridges a gap between the university studies and the real job. This is a real job. And some of us, like me, are working since more than 30 years already at the museum. We don't get rich like that. So if there are no students here today, I think that might be disappointed that you will not get very well known and very rich by this work, but it's an interesting work that keeps you young in your brain because you have to be so flexible. And you also need quite a lot of patience, especially when there are lots of visitors, like in the Christmas season, for example. The whole house is extremely crowded, or in certain weekends. And then sometimes there are really funny questions. There are always lots of questions. I remember once we were in the insect hall, and one student that seemed to be a bit cheeky and a bit bored at the same time asked me, "Oh, sir, do you have Mutilla Europea as well in your collection?" And I said, "Yes, of course. Just turn around to the other side of the display case and you will find it." Really? You know everything here in this hall? I said, "Well, quite a lot." But of course, it was a good trick, because if he had asked me about some South American or African insect and given me just the Latin name, I had no idea what to say. But since I know this animal, it's the velvet ant, I knew exactly where to find it. And another sample, another anecdote. One day when we worked in the former children's hall with open programs like in Deck 50 now, a little boy came with a stone and packed a newspaper, and he unpacked the stone, and the stone looked like this, with a hole in the middle and cut and polished. And it was obvious that this was a stone axe from the Neolithic. And I asked him, "Where did you get that?" "Oh, I found it when I was on holidays with my parents, and I wondered why there is a hole in that stone, and so I took it and I wanted to know something about this stone." So then actually we went with this boy to the prehistoric department, and the boss of the prehistoric department explained that this is a Neolithic stone axe, and gave him some information. And then of course the boy said, "Oh, may I keep it?" And he said, he opened the drawer and there were dozens of them. And he said, "Yes, of course, you can keep it." And I'm sure this boy, that's probably now an adult man who has children himself, still has this stone axe. So you see, there are sometimes very funny experiences, some very funny anecdotes to tell. So this was one of the common school classes. Common school starts with 10 years and goes until 15 in Austria. We call it now Neue Mittelschule. Before it was called the Hauptschule. And this is actually a school type where the pupils normally don't get any experience in genetics. They know that living being consists of cells. They know that there are nucleus inside the cells, and that inside the nucleus there is the DNA. And that's all what they learn about genetics. But there are some very engaged teachers, and they come with these classes. They prepare the classes with a little bit more of genetic knowledge, and then they came to our Mendel projects. As a general rule, you can say engaged teachers produce engaged and interested classes. Very often, if I greet the teacher, I know already how the class will be, and normally I'm right. Because even if you're very engaged, sometimes all your engagement fails if you have a class that's not interested or extremely tired, for example. So these are some of the pieces that my colleague Jessica photographed, and she was kind enough to send me the photos. And in this case, you have a sudden outcome. You see that the children liked what they do, how colorful and funny these things are, whether they obeyed the genetic rules or the evolutionary rules or not. At least they had fun, they learned something. And if there is time enough, we give the children time to present their pieces, to talk about them. Why do they have eyes? Why do they have 5 eyes or 2 eyes or no eyes? Or why do they have wings or tails and these things? And sometimes they really invent interesting stories. So, on the other hand, you very often don't know what you provoke with your guided tours. If you can somehow help them to understand nature better, to love nature more. I must say, during my career, I met some children with whom I had contact, then later on for some time, or with one family I still have very good contact. And you can actually influence children or youngsters with your guided tours. Maybe just this one guided tour that you gave today will make them be so interested in science that they later on decide to become scientists or to contribute in a project of protection. So my second leg, as I said, is the work of in the evening school, where I'm also working since 30 years already. Genetics is one part of this format. It's courses for young adults between beginning of their 20s to the mid-30s who missed to do the final exams at school for whatever reason, and then later on they decide now they want to do it, and they book one of our courses. In these courses, they learn about zoology, botany, ecology, the human body, cytology, and genetics, ethology. So quite a lot of subjects within one semester, within 1.5 year. So we have just 9 hours for genetics. And once again, we have to start at 0, because not all of them have learned much about genetics. There are some that come from the common school and they know just the extreme basics. And of course, 9 hours is not enough, especially if there are questions, if there are discussions. There should be at least 12 hours. And of course, we also need some time to answer questions. There are almost regularly these questions, how to clone a dinosaur. And yeah, everybody knows these Jurassic World films. And of course, we have to repeat the stuff because we have to prepare these youngsters for the exams. And as a consequence, genetics stay quite close to the basics. Well, we can surpass Mendelian genetics a bit, but there are these extremely interesting questions. For example, how do colors get inherited in cats, and colors plus patterns, because these are 2 different genetic traits, and I'm a cat person, so that interests me quite a lot. And the main problem is not that science is too difficult to talk about it, but there is never enough time. What's about all these other interesting genetic subjects like Neanderthals, epigenetics? How can I clone a mammoth? Or well, I can't, I've never tried. And I think we should better save the species from extinction that we still have than cloning extinct species. Or the 7 daughters of Eve, the 7 different genetic lines that probably exist in Europe. So this is one of these teachers who are very engaged and came with their common class schools to our Mendel project. In schools, they face different problems than we do at the museum or in the evening school, because in the museum there's always the teacher with the class, the teacher surveys the class so that they behave well. Otherwise we can bark at the class as well if they don't behave well. There are still classes with almost 30 pupils in Austria, although the government promised some years ago there will only be classes with 20 pupils. That would be almost paradise, but unfortunately this did not come to being yet. Then, of course, there are disciplinary problems, especially with children between 13 and 15. Their hormones are working stronger than their brains. And especially in Vienna and surroundings, we have lots of children that come from other nations. They speak different languages. Sometimes there are school classes with children from all different nations, but no Austrians in one school class. So there are 7, 8, 9, 10 different languages needed. And for example, in one of these classes, we had a girl from Syria who came just lately and did not speak more than 3 or 4 words in German, and so she needed a translator. Fortunately, there was another Arabian girl in the class who could translate for her classmate. And sometimes we help out with English. One of my colleagues speaks Serbian and Russian, and so we can manage. Because especially in an activity like microscopy, if you don't understand one word German and all the explanations are in German, you feel simply bored. Sometimes in schools there is also a lack of interest, and finally there are not enough biology lessons. And finally, one very interesting thing is I have a collection of some 50 biology school books at home, and I looked them through. Many things in genetics stayed the same over an extremely long period. For example, one old school book from 1943, well, not the best days for science as we have heard yesterday, but there's this example with the different forms of maize grains, and in my school book from 1976, it's almost identically the same example. Or one school book from Karl von Frisch, the famous bee researcher, 1953, and again 1976. It's almost identical pictures. So genetics didn't move on very much. Very strange. Oh, once again, 1943, the fruit flies, 1976, the same images. I don't say that these examples are bad or not interesting, but haven't we learned more? And finally, one very modern school book that I bought just a few days ago, very interesting modern samples about lactose tolerance and intolerance, for example. It's a topic that's very interesting to lots of people because they suffer from lactose intolerance or the genetic bottlenecks, for example, in the, as an example, the European bison. So there are definitely not enough biology lessons and biology excursions. And what worries me even more than the lack of knowledge in genetics, maybe that's not too welcome if I say it in this group, but it's important for me, people hardly know their indigenous species anymore. They are hardly capable of distinguishing a stork from a pigeon. And there is a serious lack of knowledge in biodiversity, but we need a knowledge in biodiversity for the understanding of nature, for the love of nature, for the respect of nature, because as it's said very often, you can protect only what you love, and you love only what you know. So we know that we know very little, and it's definitely not enough. We need more biological education for the benefit of humans and for the whole biodiversity of the planet. So there is a little bit of biodiversity from the last years that I photographed. Thank you very much for your attention. I hope I could give you some impression of the work of a pirate-like freelance biologist. Thank you.
Thank you very much for your wonderful presentations and inspirative presentation. I think it's really a great opportunity here that we can share your experiences from the museum and from schools and here with our experiences, which is really amazing. I really admired and like how you used interactive methods in your programs. And like Mendel, we have actually the same experience from our Mendel Festival, which you also experienced with us. In our case, it was an actor, a famous Brno actor who presented Mendel, and it seemed to be very successful and well accepted by the public and families and children and so on. And also I really enjoyed your interactive programs with the open questions, which made space for inspiration and creativity, which we could see in your programs. So thank you very much. Are there any questions or points for discussions from others?
Hello, I liked your talk very much, and may I have a question about the Nature Museum? Which languages are you able to have the lectures?
Always in German and sometimes in English. Sometimes we get guest speakers from the United States or from the United Kingdom or other countries. They know English then. Thank you. But the principal language is German. Unfortunately, we don't present speeches in Czech.
That was my second question, so already answered it.
I personally think that we should not only teach English or a Roman language like French or Spanish. In Austria, it should also be one Slavic language. But yeah, see all these problems with school classes.
Because we regularly visit Austria with children and visit different nature departments. And there are very nice programs for kids, but only in German. And if they speak only English, it's not always possible to have it in English there. So that's why I'm asking.
The programs at the museum can also be given in English. That's no problem. And like I mentioned, one of my colleagues at least speaks Serbian. I don't know if that would be of much help for you, or if you know any colleagues in Serbia that would like to come. I think she could do the tours in Serbian for the children.
Okay, thanks.
Thank you very much for your presentations. I must say that the Natural History Museum is one of my favorite museums in the world, so it's very nice to hear something more about that. One of the things I was wondering, because that's more with respect to biodiversity, my experience, for instance, was that one time I went to a national park in the United States, and I heard some people complaining that they didn't see all the nice animals that they would normally see in the documentaries, for instance. So I was also wondering, when you go to a museum with the children where they see all the nice butterflies, but in reality going outside is different. And it's more a general question how that is in Austria, whether children also come in contact with nature. Do they go into the field? So that's not so much something for the museum, but I think that's also an important aspect.
You're absolutely right. There are lots of people who come to national parks and then they say, oh, there are no animals there. I've worked for many years in the Danube National Park, but we never encountered the red deer or the wild boar, and beavers only occasionally. So you have to keep to the smaller animals very often and just talk about the big ones, or you see at least the tracks or the traits. And about children and contact in nature, the curve is going down. And that's thanks to all the digital activities they have. everybody is sitting with the cell phone in the underground. I'm actually happy if I still see children that are reading books. And I see it as a very big danger for nature that the young generation is so much interested in the digital world, and they have by far not enough contact to the real world, to real life. And this danger is the danger with the modern digital media, to me, is by far bigger than just the concurrence of television, for example.
So I'm not from Austria, I'm from Taiwan originally, but I just like to add a little bit of comment to that. Was when I started my university years in life science department, it was one of the first life science departments that was established as a program, degree program. But following that, programs in zoology, botany, so on, started to close down and convert into life science programs, which is mostly and exclusively focused on molecular biology and cellular biology. And it does feel like our fieldwork component exposure with the meso level world is decreasing even for biologists. And I'm quite concerned about that.
But on the other hand, if you manage as a teacher to go with your class out to nature with an interpreter or without an interpreter, they are normally flabbergasted and extremely happy. Not just because there is water and dirt and stones, it's about the animals and being outside. There are lots of children in Vienna that probably have never been to the forest, although the Viennese forests are just at the doorstep of Vienna. Yeah, the Wienerwald, exactly. And that's actually very, very worrying. That's why I said we need more excursions as well, where you go out with the children. And the younger the children are, the better it is. If you start with some youngsters of 14, 15 years, it's almost too late. But if you start with them in the kindergarten, in the crèche, or in the primary school, it works, and you can still keep that interest, maybe with this interruption of the hormonal years.
Can I add one other remark that's more
Please.
on the educational part? And you mentioned Greg Redick and also Kostas Kampourakis. My opinion is that they are quite extreme, maybe not in the for philosophy of sciences, but certainly in the genetics curriculum. It's quite an extreme position that they take. I think basically my opinion is that they make some kind of caricature of the genetics curriculum, and then they attack that. And to some extent, they want to remove beans or peas from the curriculum. And I really think that would be really a great pity. When I I was a student, actually, I was really fascinated by the story of Gregor Mendel, and that, therefore I became a geneticist. I think it's a great story, and we need scientific heroes. It's like we need Darwin for evolution. So I hope that's not your only leading point, or how to say that.
Yeah, it was one of the starting inspirations, but as you can see, we incorporated as much of the different dimensions of Mendel in the program as we could, especially with the interactive theater, because as you said, and us being immersed in Mendel this entire year because of this project, where we learned more and more about how incredible so many aspects of his story was. I am personally impressed, not just by the bee house, that he built a literal house for bees, but also the tornado paper, which is incredible theoretical detective work of him standing there and then immediately in deducing the parameters of the tornado and realizing that it breaks a commonly assumed law of rotation, like just like that, standing on the spot. It's just absolutely incredible. So In the Mendel Symposium that we organized at the Konrad Lorenz Institute a few weeks ago, we also invited Greg and Costas, amongst others, as well as Brian Donovan. You probably know his program on humane genetics that he's operating from Colorado.
I'm a plant breeder, so I don't know.
Yeah, so the three of them talked about this at the symposium as well. And we also had Eva Jablanka, who is one of the main advocates of extended or inclusive inheritance. And one of the conclusions was how important it is to go back to Mendel himself and then go through what kind of scientific processes were involved at the time in the context of the science and how instructive this is. And so to some of us, the issue is not so much that there's anything wrong with Mendel or Mendelian genetics, but just stopping there. So if that's the first and only point of entry into the world genetics, what is the risk of that? And whether there are ways that we can expand on it.
I don't think that is true actually in the curriculum. So I think it's a caricature. So I don't see what's wrong with the caricature, with the curriculum.
Can I comment on that? I'm Barbara Fischer. I'm a co-author of this textbook, which Peter projected towards the end. So I know a little bit about the biology curriculum in Austria, and I think that extrapolates to Europe. So yes, the curriculum is kind of up to date, but the curriculum is not the reality. Biology textbooks are the reality of what is being taught. And Peter explained quite nicely that the biology textbooks are somewhat lagging behind the science. So there's these cycles of publication of new textbooks are very, very slow. So what's being taught in high school usually is the research from 30 years ago. And in terms of genetics, despite the fact that the curricula are maybe more open and wider, they leave space for contemporary research. What's being taught in school in terms of inheritance ends with Mendel. And I think there's room for Mendel, absolutely. And we should teach Mendel and we should teach the piece, but we should teach that this doesn't extrapolate to understanding evolution. Like Mendelian genetics isn't the full story. And these were very, very important experiments.
I think you also have to be very careful with all these new developments. For instance, epigenetics, that clearly has been a hype. And it's certainly the inheritance of epigenetic marks and the importance for the evolution. Now people think it's not so important anymore. So you also have these hypes in science.
Yes, but when I was involved with these programs at the Natural History Museum, we got those questions actually from the instructors, from Peter's colleagues, when we went through the different examples. But which ones of these examples are actually, where are the traits that are determined by one gene? And I said, well, there are almost none. There are Mendelian traits, there's databases of Mendelian traits.
Many
but
traits are polygenic. That's
yes,
what you
exactly.
say.
That's what that's the
we know since Fischer. That's what we know since the 1920s. That's also being taught. I was taught that when I was a student.
I think so, but I think there's little focus on the fact that that most traits are polygenic and that there's lots of interactions between development and genetic influences.
I think if you explain something, then you start with something simple. So that's why you start with a single Mendelian gene.
And I totally agree with you there, but I think we shouldn't end there. Right now, what tends to happen is the time is short and the subjects to be covered are many. And then Mendel's piece get explained, and then that's where it ends. And I think that's the problem. I think we should continue, and we should tell the students that inheritance is more complex than that.
Well, that's true. I think what's also important, and that's the thing that I'm often missing, is that, for instance, the importance of genetics for plant breeding and things like that. It's not only diseases, but it's also what we eat, for instance, things like that. Yeah.
Thank you.